We're now continuing with the nervous system, specifically the sensory division for the special senses. That would be, of course, smell, taste, our ability to hear, equilibrium is a sensation, as well as vision. So starting, of course, with our senses, they begin with a receptor. So these are distinct cells of the nervous tissue that, of course, detect changes in the environment, whether it be internally, such as, say, blood pressure, pressure in your lungs, the stretch of your stomach, or externally, pressure and pain that's in your skin, stretch in a muscle, for example, more superficial locations. And we have various types of receptors, you notice, all around the body. So we'll begin by introducing some of the general sense receptors that are located all throughout the body. So starting here, what's known as a nociceptor. These are receptors to detect specifically damage to tissue, and we interpret that as pain, so injury and painful stimulation. Thermal is in heat, so we have our various types of temperature receptors, whether it be, of course, hot or cold. Mechanoreceptors, they detect pressure, so force being put onto something, and we interpret that as a physical touch. Proprioceptors, these are now located, you notice, in the appendages of the body. And proprioception, this is awareness of the position of our appendages. So we know the position of our arms and legs as a sensation. And chemoreceptors, of course, responding and detecting, of course, some unique chemical. So there's a distinct chemoreceptor for a distinct chemical or class of chemicals. And again, these are general types of receptors found all throughout the body. So we could feel pain, of course, in multiple locations, temperature. We've already learned examples of mechanoreceptors, such as the tactile corpuscle in the skin or the lamellated corpuscle of the skin. And just a few examples, here they are. So going way back to the integument section. And these are examples of mechanoreceptors. Something touches our skin, puts pressure on it, and we now interpret that as a touch sensation. Example of chemoreceptors, here especially for the levels of carbon dioxide that's in our blood. Since carbon dioxide has a direct impact on our body's pH, as another example of a chemoreceptor. Each receptor has its distinct receptive field, the area in which it, of course, can detect stimulation. And just some examples of various types of receptors. And just an interesting note, especially when it comes to nociceptors and our sensation of pain, when it comes to our internal organs, our perception of pain doesn't always match up with the location. So just as an example, we're looking at what's called a visceral sensation. Again, sensations from our internal organs. And we can think about the placement of the heart, and especially more common in men, getting this numbing kind of pain sensation down the left arm when there's a heart attack, injury to the heart. So getting back on track now, looking at the receptors, these are the cells that, of course, detect the various type of stimulation, whether it be hot or cold or pressure or pain. So there's various unique cell structures to receptors, and thus their specific function. And each receptor, of course, is adapted to be specific to some unique type of stimulation. So again, whether it's a receptor that detects an increase in temperature, a receptor that detects light, a receptor that detects pressure. And the principal job now of a receptor is to change that incoming initial stimulation, whether it be light or pressure or pain or a certain chemical, and ultimately change that initial stimulation and convert it into an electrical chemical impulse. And this is truly what our nerves, our neurons, our nervous system can now respond to. However, in order to have that transduction, the initial stimulus needs to be strong enough to, of course, stimulate the receptor and lead to that conversion. And in fact, our receptors can adapt. So we have this description of adaptation. So upon initial stimulation, initial exposure, our receptors are much more sensitive and can be easily stimulated. But you can imagine with prolonged stimulation, we now start to have this tolerance, this adaptation. So to keep that same level of stimulation active, there will need to be an increase in the level of stimulation. So a real simple example of this, a good one would be smell. So if you're in your kitchen cooking, so you've been there for some time, and of course your sense of smell will adapt to what you're cooking. So the stimulation may not be as strong for you who's been there for a while, but someone else who just walks into the room after you've already been cooking, they can now really appreciate that scent. So that'd be one example. And to see how a receptor works here now in this illustration, a mechanoreceptor, how does pressure get transduced into an electrical current? Well, here's an example of this cell membrane. You notice the phospholipid bilayer, and then we have this channel here. And the fact that we put pressure on the cell, that pressure physically opens an ion channel. And now we have this current flowing in. And at this point, we have that electrical current. And now we could stimulate the neurons. Or an example of a thermoreceptor, well, that increase in temperature, and temperature, of course, has a huge impact on our physiology, so affecting this enzyme's function, and in this case, thus opening up this ion channel based on temperature. So in some way, the receptor is responding to the initial stimulation, and that leads now ultimately to a electrical current, a voltage. So in all cases, it's the receptor that's unique in some way for a certain type of stimulation. It'll transduce and convert that original stimulation truly into an electrical current. And the next step is to signal to a sensory neuron. And at this point, of course, sensory neurons are now sending their information towards the central nervous system, ultimately the brain, and we could respond to and perceive these stimulations. So now getting back to the special senses, the special senses are now just simply a large cluster of a certain type of receptor in a dedicated special sense organ. So taking a look at the 5 special senses, our sense of smell, olfaction, this comes down to the number of chemoreceptors, chemical receptors, that are right in the superior portion of the nasal cavity. So we breathe, and the various chemicals that stimulate these receptors we interpret as smell. The same for taste. Gustation comes down to chemoreceptors that are on our tongue, even in our throat as well. And the chemicals, the material that we ingest on the surface of our tongue especially, we have our taste buds containing chemoreceptors, and then we interpret that as flavor and taste from what we consume. We notice now the inner ear. The inner ear is a dual sensory organ. So of course we use our ears to hear sound. That's the auditory function, interpreting sound sensation, but also equilibrium, just our position and our motion of the head, which is vital for balance or equilibrium. And of course, the last one here, looking at the eyes and the detection of light, and we interpret this as, of course, our visual sensation. So we'll take a look at these various organs and definitely describe the types of receptors that are in these various locations. So the first one, really the simplest of these special senses, we see the anatomy of the nasal cavity, and we've covered this with the respiratory system. So as we breathe in, And you notice here, right at the roof of the nasal cavity is where we have the cluster of chemoreceptors. And ultimately, these signal to that first cranial nerve, the olfactory nerve. And of course, we interpret this sensation as smell. But it truly comes down to the chemoreceptors. And that's what we're seeing here. A cluster of chemoreceptors right at the superior edge of the nasal cavity. So in this small, compact area, we have our concentration of chemoreceptors. And ultimately, we interpret this information in the temporal lobe of the brain. And one note about our sense of smell, this is one sensation, and there's a few other examples, that has no interaction with the thalamus of the brain. So the sensory information for olfaction does not pass through the thalamus as many other sensations do. And this is actually due to the fact that the olfactory tract itself, which is the correct term, what we typically call the first cranial nerve, is not truly a nerve, but an outcropping of brain tissue, hence the more correct term, olfactory tract. And that olfactory tract, of course, leads in and carries that information itself right into the temporal lobe, right within the cerebrum. So it doesn't need that thalamus bypass. And for sense of smell, again, we'll just go back to the view. Cluster of chemoreceptors, and just based on the sense that we inhale, the chemicals specifically, binding to those receptors. And again, we can see that outcropping of brain tissue that we now commonly refer to as the olfactory nerve, but with the more correct term there, olfactory tract. Gustation, our sense of taste. Again, chemoreceptors is the basis of this sensation. So this comes down to ultimately the food that we consume, and more importantly, what gets dissolved into our saliva, which is key. We need moisture, we need saliva in order to have our function for sense of taste. So of course, these gustatory receptor cells are just simply the chemoreceptors. They're found in the taste buds of the tongue, as well as down the pharynx. And taking a look at the surface of the tongue, we have this view here to see the various sets of papillae. So you notice here from the anterior tip of the tongue, and also down the medial section, we have these fungiform papillae. You notice more to the lateral aspect of the tongue, both sides, of course. And then ultimately, the posterior circular wall would be that translation there. The various sets. And just seeing the full view of this figure. So we're seeing the location of these various types of papillae, but more importantly, you notice along the sides of a papilla is where we have the location of the taste buds, which themselves are now filled with the chemoreceptors, the gustatory receptor cells. So clustered within a single taste bud, we could have a number of these distinct chemoreceptors, these gustatory receptor cells. And again, depending on the foods that we consume and the material that gets dissolved into our saliva. And dissolving into the saliva is key because the taste buds are located along the sides of the papillae, and that saliva drains down the sides of the papillae, exposing it to the taste buds. And just a larger view of it here. So we need that moisture to deliver material to the taste buds, the location of chemoreceptors. So we see that there's a diversity in the types of chemoreceptors. So just simply put, what's known as sweet receptors responding to sugar. So just simple sugars like fructose will make something have the perception of being sweet. Makes perfect sense here. Salt receptors detecting sodium. Acid is what makes something taste sour. And the opposite of that, the opposite of acid, so something that's alkaline, higher in pH. So that's interpreted as a bitter taste. An alkaloid. We have water receptors. So if you had absolutely pure water, it would taste like water. And finally, there's what's known as umami is the term. And umami receptors specifically are responding to glutamate. And most commonly, you've probably heard of glutamate. That's the G in monosodium glutamate. And it's described as a very savory flavor. And if you're unsure if you've ever had umami flavoring, which you've had the little seasoning packets that come with the ramen noodles, MSG, or you've had a bag of Doritos. You might have one with you right now. Just go ahead and look at the label on the back of Doritos, MSG. So now we're moving on to the ears. As we stated from the beginning, the ears are a dual sensory organ. So for both our ability to hear, which we'll tackle first, as well as our equilibrium sensation. So we'll start out with, of course, the anatomy of the ear as a whole. And there's 3 distinct regions of the ear, and all are required for our ability to hear. So there's just simply from the superficial surface, right, external ear, a middle ear, and what could be called inner or internal ear. So we'll start at the surface, and what you commonly refer to as your ear on the side of our head, the auricle. And this works as simply as a funnel to channel sound waves ultimately down our external acoustic canal and ultimately to reach the eardrum, the tympanic membrane. So beginning at the auricle, we have our external auditory canal, and of course this leads inwards towards the eardrum. The skin, of course, lining this area could contain, of course, various amounts of hair growth, especially that intermediate hair, and as well as the skin gland known as the cerumeus gland, the production of that protective earwax. And that's the importance of both the hair and the wax can protect the tympanic membrane itself. So we could trap any particulate matter with the hair or the wax before it reaches the very delicate tympanic membrane. And the tympanic membrane itself is an extremely delicate piece of skin. So just a very, very thin, delicate piece of skin at the end of our external auditory canal. And a sound wave will now trigger a vibration of the eardrum. And ultimately, the pattern and strength of this vibration is what we detect and interpret as a sound. But truly, from this point on, we're just detecting the vibration inside of our skull, and we interpret that vibration as a sound. So we can see that visually here, the external ear, and again, the auricle, just channeling the sound waves from the environment in. So you notice here there's multiple names for the ear canal, okay, that are acceptable. Auditory canal, acoustic meatus. And then ultimately we trigger that vibration in the tympanic membrane. So from here, you could already see that there's a connection on to the middle ear, and that vibration will pass through these very tiny little bones. And then finally, leading into the inner ear, this is now the hollow cavity inside of the temporal bone. And that's where we find the cochlea, the area involved in ultimately detecting these vibrations. So vibration waves pass through, and it's in this area where we have the receptors. So we'll continue with the middle ear, and we just pointed out those very tiny bones, collectively known as auditory ossicles. And individually, there's the malleus, incus, and a stapes. They're found in this order. So the first one, the malleus, is attached to the tympanic membrane. That vibrates against the incus, and in turn, the incus vibrates against the stapes, which itself is attached to the oval window. This is now the first part of the inner ear, the first structure of the inner ear. So ultimately, the middle ear itself contains these three very tiny bones of the skeleton, the ossicles, as well as it's a air-filled chamber. So we see that connection between the eardrum and ultimately to the inner ear. And these bones, these ossicles, not only transmit the vibration, but they also boost and strengthen the vibration. So our tympanic membrane, the eardrum alone, does not trigger sufficient levels of vibration that the inner ear can detect. So the vibration from the tympanic membrane is amplified by the ossicles. And now at this point, the inner ear can detect these stronger vibrations. However, to protect our hearing, to protect the structures from damage, in response to extremely loud sounds, as you can imagine like a gunshot, a very, very loud sound, there are muscles, skeletal muscles specifically, in the area of the middle ear, and these muscles will contract to reduce the vibrations in response to a loud sound so that we don't have extremely strong vibrations and thus possible damage to our ear structures. Another portion of the middle ear, there's many, many names for this: pharyngotympanic tube, eustachian tube, simpler term, auditory tube. And this is now a connection from the middle ear, and it leads to the nasopharynx. And the purpose of this auditory tube, it's typically closed off, and it opens briefly to allow for an adjustment of pressure. There's the pressure of the environment that's superficial or external to our eardrum. And then there's a pressure internally inside of our head, specifically between the pharynx and the middle ear. And this auditory tube will open to allow for a balance of the pressure. So a good example is, you're in an airplane, you're in that pressurized cabin. So there's the pressure inside the airplane that's, of course, externally to your ear, superficial and external to your eardrum. And there's a pressure, of course, that's within your skull and going up to atmosphere in that pressurized cabin. You probably heard of chewing gum to help you make that balance. But typically the auditory tube is closed off. And just a quick note about young children, due to the size of their head, in young children, the auditory tube is shorter and has a more horizontal position, whereas in an adult, the auditory tube is angled down, slanting at an angle, and we'll point that out. But in children, it's shorter and more horizontal, meaning it's easier for infection from their nose to pass into their middle ear and cause an ear infection. So we can see that here. So this illustration is showing the angled adult structure for the auditory tube and how it leads from the nasopharynx, leading right up to the air-filled chamber of the middle ear. And as we already described, the principal function of the middle ear is to boost and transmit vibrations from the eardrum to the inner ear. And again, multiple names that are all acceptable. Good example here, eustachian tube is the eponym, so named for a person. Pharyngotympanic tube, just extremely descriptive term, connects the pharynx, nasopharynx specifically, to the tympanic cavity, the space of the middle ear. And just a close look at the ossicles themselves. We'll see these in lab. Malleus, incus, stapes. Probably heard the common terms: hammer, anvil, stirrup. So now ultimately, it's that stapes that connects now to the inner ear. And the inner ear itself, again, is a hollow cavity within the temporal bone. So one of the many bones of the cranium. And specifically, the sinus cavity within the temporal bone is known as a bony labyrinth. And now inside of that bony sinus, there's a soft connective tissue structure known as the membranous labyrinth. And it follows every curve and contour of the more superficial bony labyrinth. And this entire inner ear, again, has two sensory functions, but we're going to look specifically at the cochlea portion of the inner ear, because that's where we detect these vibrations that we interpret as sound. We'll come back to the remainder of the inner ear in just a moment, but the remainder of the inner ear, this is the portion used for our equilibrium sensation. So continuing with the cochlea specifically for now, we notice that ultimately comes down to pressure receptors, mechanoreceptors. And the reason being is vibration that was detected by the eardrum, sound wave vibrates the eardrum, the vibration passes through the ossicles. Specifically, that third ossicle, stapes, vibrates against the oval window. The inner ear is now filled with different types of fluids. And ultimately, this now wave of vibration that's now passing through the fluids is ultimately going to now distort what's known as these hair cell receptors. And the hair cell receptors themselves, with their stereocilia, are now bent. They're distorted due to the pressure wave passing through the fluid. So it's ultimately these mechanoreceptors, known as the hair cells, that pressure and vibration that the fluid puts against the hair cells, that activity of mechanoreceptors is ultimately what's going to stimulate our eighth cranial nerve. And ultimately now we interpret this vibration and pressure as a sound. So just describing the inner ear again, our hearing, our auditory sensation comes down to mechanoreceptors for both sound as well as the equilibrium portion of the inner ear. They're both based on mechanoreceptors, and that comes from the motion within the fluid. Again, the more superficial sinus cavity of the temporal bone is known as a bony labyrinth. And the deeper connective tissue layer is the membranous labyrinth within. So we can see this here again. We're looking at the inner ear, the sinus cavity of the temporal bone. We're looking specifically at the spiraling portion of the cochlea. So you notice here spiraling around 2.5 times. You can see that entire apparatus, of course, located within the temporal bone. So now we have this figure here. We're able to see the entire inner ear. And you notice in the tan shading, the superficial bone tissue sinus of the temporal bone, the bony labyrinth, all throughout here, wherever you see the tan color at the surface. And now deeper in, here illustrated in blue, following every curve and contour. This area highlighted in blue is now all the internal connective tissue membranous labyrinth spanning within. So of course, we're going to stay focused on the area of the cochlea for hearing. So now if we have a slice through the cochlea, we can see that as it spirals around, there's a set of 3 chambers, and the same 3 chambers are just spiraling around and around, as I mentioned, 2.5 times. So if we zoom in on just one set of 3, that's how we get this view here. So you can see the spiraling of the cochlea with its 3 chambers. Zooming in on just one set of 3, and we see these various areas known as the vestibular duct, in the middle cochlear duct, and a tympanic duct, separated by various membranes. And in the inset right here in the box, we notice there's the stereocilia of the hair cells. So these are the mechanoreceptors, and the pressure that comes from the vibration will ultimately stimulate those hair cells. So going back to the point that the inner ear is filled with 2 types of fluid, that vestibular duct and tympanic duct, they share the same type of fluid known as perilymph. The reason being is, we notice here from the oval window, and the vibration wave will pass through the vestibular duct, just the alternate name there for vestibular duct, and that vibration wave passes through the fluid. And this is showing the hypothetical situation if you were to unravel and uncoil the cochlea. But ultimately, the vestibular duct and the tympanic duct are connected to one another. And for that reason, they share the same fluid. Whereas the middle chamber in the center, the cochlear duct, and here's again an alternate name, scala media, the cochlear duct in the middle, has its own distinct fluid known as endolymph. And here's just showing that idea. A wave of vibration, of course, passes through and around through the vestibular duct, and then ultimately it leads right into the tympanic duct, and the vibration wave can pass out of the cochlea, and that, of course, allows us to first begin hearing a sound due to the vibration, and then, of course, the sound, of course, dissipates, and we stop hearing the sound as the pressure wave exits. So ultimately, those stereocilia, the hair cells, receptors, their job is to signal to a sensory nerve, and that happens to be the 8th cranial nerve you notice here. And now we're taking that sound stimulation that started as a sound wave. It was now converted to a vibration, and now mechanoreceptors detect the pressure of the fluid, the vibration occurring within the cochlea, and ultimately we interpret our sound in the temporal lobe of the brain. So now we'll continue with the special senses, and we'll move on to the last 2. So staying with the inner ear, once again, as we described, the entire ear function is based on mechanoreceptors, so pressure receptors. But now for equilibrium, we're looking at the area outside of the cochlea. So the vestibular apparatus that includes the semicircular ducts, the ampulla. And starting with these 2 areas specifically, the semicircular duct structures, the ampulla, these now allow for the sensation, what's known as dynamic equilibrium, or just simply rotation of our head. So truly, we could tilt and rotate our head in various planes, and that position of our head is used for our equilibrium. So an awareness of the position of our head, our place in gravity, And we use this sensation to then coordinate our balance, coordinate the muscles involved with balance. Also, more mechanoreceptors within the inner ear, specifically the utricle and the saccule portions. These now detect the sensation, what's known as static equilibrium, or it could be described as linear motion. So we're aware of motion, if we're moving upwards vertically or downwards. Think about going in an elevator would be a good example. So we have that sensation that we're moving in our place in gravity, but not only vertically, but horizontal motion as well. And we see again in terms of equilibrium, all of this sensory information for equilibrium is actually now diverted to the cerebellum of the brain. And the principal function of the cerebellum is coordinating our muscles to maintain our balance, or coordinating our muscles to maintain our equilibrium. So we can see, of course, that same inner ear region that we were just describing for sound. But again, it's now this portion here, the vestibular apparatus, that's used for equilibrium sensation. And we notice the three looping areas, the semicircular canals and semicircular ducts. And as we rotate our head, as we're tilting our head, say to imply yes, tilting our head, shaking side to side as if we're implying no, and tilting our head laterally. So as we rotate our head in either of these planes, ultimately the fluid endolymph within the membranous labyrinth specifically, that rotation will allow for that pressure to be put directly on the ampulla. And again, these mechanoreceptors now interpret that pressure, and we interpret that as, of course, rotation of the head. So the area here in the box is the equilibrium portion of the inner ear. And we've already discussed, of course, the cochlea is the auditory portion of the inner ear. So it's this dual sensory structure. So to finish the special senses, we're moving into vision, sight with our eyes. And you notice here, a large percentage of all of our body's receptors. So of any type, a large percentage of all of our body's receptors are found in just this organ here, the eye. So we'll see that the detectors, the receptors for light in the eye, especially, number in the millions. So there's millions and millions of photoreceptors. But of course, keep in mind, the eye has other sensations, pressure and pain, the general sensations. The special sense receptor is the photoreceptor. So now moving forward with the photoreceptors, they come in two varieties. So there's what's known as the cones. The cone photoreceptors, and it really comes due to their conical shape. There's three subtypes, and what's simply known as the red, blue, and green cones. And the cones, of course, allow for color perception, as well as giving the much finer detail for our vision. Whereas the rods lack color detection, but they're actually more sensitive to light. So we'll come back and compare these two classes of photoreceptors. Looking at the gross anatomy of the eye itself, the eye has three tunics, three layers would be the term. So three principal layers. And that's what we see here as the fibrous tunic, vascular tunic, and it could be called neural tunic or nervous tunic. So starting at the surface, there's a superficial fibrous tunic that only has two components. The majority of the fibrous tunic, the external sclera, and you see that simple description there. And you can see blood vessels, of course, running through the surface as well. So that thick, strong external layer of the eye. However, at the anterior portion of the eye, there's the cornea, and the clear cornea that allows for light to enter the eye. But also, this is an important structure that's adjusting the focus as light enters your eye. So typically, any issues with the shape and structure of your cornea may lead to the need of corrective lenses, glasses, contacts, or even now, of course, laser surgery. The middle layer of the eye, the vascular tunic, with multiple components. So the choroid region, this is truly the vascular layer, hence the name you notice. So rich in the blood supply for the eye. There's a ciliary body structure, which is both a muscle, it's both a smooth muscle structure, and it's a gland that produces this aqueous humor. The aqueous humor fills the anterior cavity of the eye, so this fluid circulating in the anterior portion of the eye, whereas the smooth muscle structure ultimately has a connection onto the lens, and this helps to adjust the shape of the lens for our far distance versus near distance, so making that adjustment. So it's fine tuning the focus of our eye. And another structure of the vascular tunic, the iris. Well, the iris contains pigmentation, so that's what dictates the color of your eye, the amount of pigmentation in the iris. Well, this is also another smooth muscle structure, and the iris, this adjusts to allow varying levels of light to enter the eye. So increasing the diameter when it's darker, or decreasing the diameter when it's lighter to adjust the amount of light that enters the eye. We notice here the lens, and it's separate from either of these tunics. The lens does not belong to any tunic of the eye, but we do see that the ciliary body and its ligaments will connect onto it. And again, the lens could be adjusted. The thickness of the lens is adjusted to focus our vision, to fine tune for near or far vision. And then lastly, the deep internal layer. So again, the neural tunic or the nervous tunic. This is now where we have the receptors. The rods and cones are located here. And specifically within the neural tunic, there's this region known as the fovea centralis. And this area only contains the cone photoreceptors. And the fovea, this is the site and the spot of our best vision. So truly, when you want to get a good look at something, you stare straight at it, and that light falls right on the fovea centralis. Whereas the area known as optic disc, this is an area that lacks any photoreceptors, hence the description, the blind spot. And the importance is the optic disc of the retina, the optic disc of the eye, then connects on to the second cranial nerve, the optic nerve. So we can see some of the superficial anatomy surrounding the eye. Again, we'll cover this in lab. We'll cover the structures in lab, but the simple idea, light enters our eye at the cornea, and the shape helps to focus it. So as light passes through the pupil of the iris, adjusted by the lens, and here's the ciliary body with its attached ligaments that could now adjust the shape of the lens. And ultimately, we want the light to be in focus when it reaches the retina, when it reaches the neural tunic. So again, we'll see more of this terminology in lab in just a moment. And here you can see a good example, looking directly at something, and the light's reaching that area known as the fovea. Whereas the optic disc, the area that lacks photoreceptors, and that leads right into the optic nerve. Ultimately, we interpret all of this sensor information from millions of receptors in our retina, and we interpret this in the occipital lobe of the brain. So to take a look at the photoreceptors themselves, these are the special sense receptors, the unique special sense receptors, just located in the retina of the eye. And again, they come in the two varieties, rods and cones. So we'll start here with the cones. So first of all, the numbers here are in the millions. I just had to abbreviate that for space on the slide. So 6 million cones located, of course, within the retina. And as we mentioned, there's the 3 variations. So responding to the different wavelengths of light, the different colors of light. And at the level of our retina, we only detect, yes, red, blue, green. And it's now our brain, that occipital lobe, that can interpret, of course, millions and millions of different shades and colors based on the pattern of stimulation. We can see that conical shape to the end of the cell, so where that name comes from. And again, the cones, in terms of our vision, it gives us the fine detail. So our ability to read the text right here on the screen, or think about when you're at the optometrist and they're showing you the Snellen chart and asking you to read the letters, or at the DMV, right? So that ability for the hot, fine detail and acuity, as well as our color vision, comes from the cones. However, we need sufficiently bright levels of light for the proper functioning of our cones. Whereas now the rods we see are much more common, and again, this is a number in the millions, 125 million rods, so extremely numerous. These are found throughout the retina, excluding that fovea centralis location. And again, the rods give no color vision. It would be better described as grayscale as opposed to black and white. So grayscale, but lacking any color discrimination. The rods do not give us the fine detail, but the benefit, of course, is rods are more sensitive to light. So even in a dim light setting, a low dim light setting, the rods are so sensitive that they can still have, of course, a degree of visual stimulation and activity despite the dim light settings. And we can see that long cylindrical shape to the rod cell structure. So this is just a view of the retina. And we can see, of course, the various types of cells, but specifically, rods and cones make up the photoreceptors, the light detecting cells, and the rest of which these other types will bring up in the histology section. So we'll finish here with this chart looking at different sections of the brain and describing just some of their principal functions, tying this right back to the special senses. So we can start with the temporal lobe on the side, the lateral portion of the cerebrum. And this is now where the information from our cochlea is interpreted in terms of sound. For our sense of smell, the chemoreceptors, of course, in our nasal cavity, signaling ultimately through that olfactory tract. And we interpret smell and sound in the temporal lobe. The frontal lobe of the brain, this is the origin point for our somatic motor function. Somatic motor. So we want to think of the control of skeletal muscle. So this is a motor pathway originating from the frontal lobe. And this is one area where we interpret taste. The insula as well. The insula of the brain is another area where we interpret taste, but as well as the frontal lobe. The parietal lobe are general sensations. So again, temperature, pressure, pain, many different general sensations. That's interpreted in the parietal lobe of the brain. And this last special sense that we just covered, of course, our eyes, our neural tunic, and all the rods and cones. Ultimately, we interpret this sense information in the occipital lobe of the brain.